So hello guys, welcome back to my YouTube channel. So in this video we are going to do NCERT reading of the chapter Biotechnology Principles and Processes. So let's start. Biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. In this sense, making curd, bread or wine, which are all micro-mediated processes, could also be thought as a form of biotechnology. However, it is used in a restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. Further, many other processes/techniques are also included under biotechnology. For example, in vitro fertilization, leading to a test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all part of biotechnology. The European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural science and organism cells, parts thereof, and molecular analogues for products and services. Principles of biotechnology. Among many, the two core techniques that enabled birth of modern biotechnology are genetic engineering, techniques to alter the chemistry of genetic material DNA and RNA to introduce this into host organisms and thus change the phenotype of the host organism. Bioprocess engineering, maintenance of sterile microbial contamination-free ambience in chemical engineering processes to enable growth of only the desired microbe/eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Let us now understand the conceptual development of the principles of genetic engineering. You probably appreciate the advantage of sexual reproduction over asexual reproduction. The former provides opportunities for variation and formulation of unique combinations of genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic information, while sexual reproduction permits variation. Traditional hybridization procedures used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. The techniques of genetic engineering, which include creation of recombinant DNA and use of gene cloning and gene transfer, overcome these limitations and allows us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. Do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in the progeny cells of the organisms. But when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become part of a chromosome, which has the ability to replicate. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. Therefore, for the multiplication of an alien piece of DNA in an organism, it needs to be a part of a chromosome, which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid autosomally replicating circular extrachromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boer accomplished this in 1972 by isolating the antibiotic resistance genes by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at specific location became possible with the discovery of the so-called molecular scissors restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. These plasmid DNA act as a vector to transfer the piece of DNA attached to it. You probably know that mosquito acts as an insect vector to transfer the malarial parasite into human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which acts on cut DNA molecules and joins their ends. This makes a new combination of circular autosomally replicating DNA created in vitro and is known as a recombinant DNA. When this DNA is transferred into Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. You can hence infer that there are three basic steps in genetically genetically modifying an organism. Identification of DNA with desirable genes. Second, introduction of the identified DNA into the host. Third, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Tools of recombinant DNA technology. Now, we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools, that is restriction enzyme, polymerase enzyme, ligase, vectors, and the host organism. Let us try to understand some of these in detail. Restriction enzymes. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl group to the DNA while the other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease, Hind second, whose functioning depended on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. It was found that Hind second always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. This specific base sequence is known as the recognition sequence for Hind second. Besides Hind second, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. The convention for naming these enzymes is the first letter of the name comes from the genus and the second two letters comes from the species of the prokaryotic cell from which they were isolated. Example, EcoR1 comes from Escherichia coli RY13. In EcoR1, the letter R is derived from the name of strain. Roman numbers following the names indicate the order in which the enzymes were isolated from that strain of bacteria. Restriction enzymes belong to a larger class of enzymes called nucleases. These are of two kinds, exonucleases and endonucleases. Exonucleases remove nucleotide from the ends of the DNA, whereas endonucleases may cut at specific position within the DNA. Each restriction endonuclease functions by inspecting the length of DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Do you know what palindromes are? These are groups of letters that form the same word when read both forward and backward, that is Malayalam, as against a word palindrome where the same word is read in both directions. The palindrome in DNA is a sequence of base pairs that reads same on the two strands when orientation of the reading is kept the same. For example, the following sequence reads the same on the two strands in 5' to 3' direction. This is also true if read in 3' to 5' direction. GAATC, and then it is CTTAAC. First one is 5' to 3' and second strand is from 3' to 5'. Restriction enzymes cut the strand of DNA a little away from the center of palindrome sites but between the same two bases on the opposite strands. This leaves single-stranded portions at the ends. There are overhanging stretches called sticky ends on each strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. This stickiness of the ends facilitates the actions of enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA which are composed of DNA from different sources' genomes. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends and these can be joined together end to end using DNA ligases. So here is a diagram. This is a foreign DNA and when we have taken vector DNA plasmid, same restriction enzyme cutting both foreign DNA and vector DNA at specific sites, and now ligase join foreign DNA to plasmid, then we get our recombinant DNA molecule, and then there is a process called transformation, and then the recombinant DNA is introduced into the bacteria or the host organism, and then it divides, and then the host divides and it is passed on to the next generation. So this is a diagrammatic representation of recombinant DNA technology. You may have realized that normally unless one cuts the vector and the source of DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonucleases results in the fragments of DNA These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move toward the anode under an electric field through a medium/matrix. Nowadays, the most commonly used matrix is agarose, which is a natural polymer extracted from seaweed. The DNA fragments separate, resolve according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. Look at the figure 11.3 and guess at which end of the gel the sample was loaded. So in the first lane, there we have this undigested DNA, and in the remaining 2, 3, 4, three wells, there is the digested set of DNA fragments that are filled. So the separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. You cannot see pure DNA fragments in the visible light and without staining. You can see the bright orange colored bands of DNA in ethidium bromide stained gel exposed to UV light. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. Cloning vectors. You know that plasmids and bacteriophages have the ability to replicate within bacterial cells independent of the control of the chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cell. Some plasmids may have only 1 or 2 copies per cell, whereas other may have 15 to 100 copies per cell. Their numbers can go even higher. If we are able to link an alien piece of DNA with bacteriophage or a plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. Vectors used at present are engineered in such a way that they help a easy linking of foreign DNA and selection of recombinants from non-recombinants. The following are the features that are required to facilitate cloning into a vector. Origin of replication. This is a sequence from where replication starts, and any piece of DNA when linked to its sequence can be made to replicate within the host cells. This sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origins support high copy number. Second is selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced into a host bacterium. You will study the process in subsequent section. Normally, the genes encoding resistance to antibiotics such as ampicillin, chlor कभी कभी ऐसे ऐसे आ जाते हैं ना वर्ड्स जो थोड़ा सा वो हो जाता है भाई, कैसे प्रोनाउंस करूँ, प्रोनाउंसिएशन देखना पड़ता है. Chloramphenicol, tetracycline, or kanamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Third is our cloning sites. In order to link the alien DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. Presence of more than one recognition sites within the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, you can ligate a foreign DNA at the BAM-H1 site of tetracycline resistance gene in the vector PBR322. The recombinant plasmid will lose tetracycline resistance due to insertion of foreign DNA but can still be selected out from non-recombinant ones by plating the transformants on the tetracycline containing medium. The transformants growing on the ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium but not on that containing tetracycline, but non-recombinants will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in the selecting the transformants, whereas the other antibiotic resistance gene gets inactivated due to insertion of alien DNA and helps in selection of recombinants. Selection of recombinants due to inactivation of antibiotics is a cumbersome में बोलेंगे क्या, एक सेकेंड. ओके, सो इट्स cumbersome मतलब डिफ़िकल्ट प्रोसीजर है. So ठीक है, cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. इसका वो होता है LAC-Z gene. In this, a recombinant DNA is inserted within the coding sequence of an enzyme, beta galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. The presence of chromogenic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. Presence of insert results into insertional inactivation of the beta galactosidase gene, and the colonies do not produce any color. These are identified as recombinant colonies. Vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria and viruses, which have known this for ages. How to deliver genes to transform eukaryotic cells and force them to do what the bacteria or viruses want. For example, Agrobacterium tumefaciens, a pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA to transform normal plant cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in this eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering genes of interest to humans. The tumor-inducing TI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector which is no more pathogenic to the plants but is still able to use the mechanism to deliver genes of our interest into a variety of plants. Similarly, to deliver genes of our interest into a variety of plants, similarly, दो बार बोल दिया क्या मैंने? Sorry guys. Similarly, Similarly, retroviruses have also been disarmed and are now used to deliver desirable genes into animal cells. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial, plant or animal host where it multiplies. Competent host for transformation with recombinant DNA. Since DNA is a hydrophilic molecule, it cannot pass through cell membranes. Why? In order to force bacteria to take up the plasmid, the bacterial cells must first be made competent to take up DNA. This is done by treating them with a specific concentration of a divalent cation, such as calcium, yes, which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42-degree heat shock and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not the only way to introduce alien DNA into host cells. In a method known as microinjection, recombinant DNA is directly injected into the nucleus of an animal cell. In another method, suitable for plant cells, are bombarded with high velocity microparticles of gold or tungsten coated with DNA in a method known as biolistics or gene gun. And the last method uses disarmed pathogen vectors, which, when allowed to infect the cells, transfer the recombinant DNA into the host. Now that we have learned about the tools for constructing recombinant DNA, let us discuss the process facilitating the recombinant DNA technology. Process of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction into nucleases, isolation of a desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at large scale, and extraction of the desired product. Let us examine each of these steps in some details. Isolation of the genetic material DNA. Recall that nucleic acid is a genetic material of all organisms without exception. In majority of organisms, this is deoxyribonucleic acid or DNA. In order to cut the DNA with the restriction enzyme, it needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as RNA, protein, polysaccharides, and also lipids. This can be achieved by treating the bacterial cells, plant or animal tissue with enzymes such as lysozyme, bacteria के लिए होता है, cellulase for plant cells, chitinase for fungus. You know that genes are located on a long molecule of DNA intertwined with proteins such as histones. The RNA can be removed by treatment with a ribonuclease, whereas protein can be removed by treatment with protease. Other molecule can be removed by appropriate treatments, and purified DNA ultimately precipitates out after the addition of chilled ethanol. This can be seen as collection of fine threads in the suspension. यहाँ पे एक डायग्राम दे रखा है. DNA that separates out can be removed by spooling. Now, cutting of DNA at specific locations. Restriction enzyme digestions are performed by incubating purified DNA molecules with the restriction enzyme at the optimal conditions for that specific enzyme. Agrogel electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is negatively charged molecule, hence it moves towards the positive electrode anode. The process is repeated with the vector DNA also. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source DNA and the cut vector with space are mixed and ligase is added. This results in the preparation of a recombinant DNA. Amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of gene or DNA of interest is synthesized in vitro using two sets of primer, small chemically synthesized oligonucleotide that are complemented to the regions of DNA, and the enzyme DNA polymerase. The enzyme extends the primer using the nucleotides provided in the reaction and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times, that is 1 billion copy are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase isolated from a bacterium Thermus aquaticus, which remain active during the high temperature. Induced denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. So यहाँ पे एक डायग्राम है जहाँ पे रीजन टू बी एम्पलीफाइड है, यहाँ पे डबल स्टैंडर्ड डीएनए है, एंड देन देयर इज डीनेचुरेशन. एक पर्टिकुलर टेम्परेचर पे डीनेचुरेशन होता है, दैट इज़ अराउंड 90 to 95 डिग्री सेल्सियस. एंड देन फिर टेम्परेचर थोड़ा और डाउन आता है, फिर एनिलिंग होता है, प्राइमर अटैच होते हैं. मतलब डीनेचुरेशन का मतलब होता है कि ये दो स्ट्रैंड जो सेपरेट हो गए डीएनए के. डबल स्टैंडर्ड होता है ना, तो ये दोनों स्ट्रैंड सेपरेट हो गए हैं. एनिलिंग में क्या होता है कि फिर एक सिंगल स्ट्रैंड जो सेपरेट हुए हैं उस पर प्राइमर आकर के जुड़ जाता है, ठीक है. और फिर प्राइमर डीएनए पॉलीमरेज का यूज़ करके और डीऑक्सी राइबो न्यूक्लियोटाइड्स को जो क्या कहते हैं उसको जोड़ता है कॉम्प्लीमेंट्री स्ट्रैंड बनाने के लिए, और ऐसे ही साइकिल चलती है. सो फर्स्ट वी हैव डीनेचुरेशन, देन एनिलिंग. एक्सटेंशन होता है हम लोगों का 72 डिग्री सेल्सियस पे, जहाँ पे कि यहाँ पे न्यूक्लियोटाइड्स जुड़ रहे हैं और कॉम्प्लीमेंट्री स्ट्रैंड बन रहा है. और ऐसे करके 30 साइकिल्स होते हैं, जिससे कि 1 बिलियन टाइम एम्पलीफाइड हो जाता है जो हमारा जीन ऑफ इंटरेस्ट है. और फिर उसको हम वेक्टर में उसको डाल के लाइगेज के थ्रू उसको जोड़ सकते हैं, तो वो फर्दर आगे रिकॉम्बिनेंट डीएनए बनेगा. फिर उसको हम होस्ट में डालेंगे और फिर होस्ट में ट्रांसफॉर्मेशन हो जाएगी और फिर ट्रांसफॉर्मेंट हो जाएगा. फिर जैसा कि पता लगा था हमें कि जो वेक्टर होता है उसके पास कॉपी नंबर होता है, तो वो फर्दर अपना जो जीन ऑफ इंटरेस्ट है वो जो होस्ट है उसके मेन क्रोमोसोमल डीएनए में इंट्रोड्यूस कर देगा और ख़ुद भी डिवाइड होगा, तो नेक्स्ट जनरेशन में भी प्रोजेनी में भी पास ऑन होगा. तो ऐसे करके हम ज़्यादा नंबर में अपना डिज़ायर्ड प्रोडक्ट बना पाएंगे. और जब वो प्रोडक्ट बन जाएगा तो फिर हम उन बैक्टीरिया को फर्दर और ग्रो करेंगे, और फिर वो बैक्टीरिया को जब लार्ज स्केल पे ग्रो करेंगे बायो रिएक्टर्स में, तो फिर उसके बाद हम उसको डाउन स्ट्रीम प्रोसेसिंग के थ्रू क्या करेंगे, उसको हम क्या कहते हैं कि सेपरेशन और प्योरीफिकेशन कर देंगे, प्रिजर्वेटिव ऐड कर देंगे. टेस्ट जैसे अगर ड्रग्स होता है तो उसमें हम लोग उसको फर्दर और बहुत सारे टेस्ट होते हैं उसके बाद ही अप्रूव होता है, तब जाके हम मार्केट में बेच सकते हैं. क्योंकि व्हेन इट कम्स टू वैक्सीन और ये सारी चीज़ें, तो ये हमारे ह्यूमंस पे डिपेंड करता है. मतलब अगर कुछ ख़राबी होगा तो इंसान ही मर जाएगा ना, तो हम ऑफकोर्स इतना बड़ा रिस्क नहीं ले सकते हैं कि किसी भी चीज़ को अप्रूव कर दे और मार्केट में जाने दे. बहुत लोग की जान जा सकती है. सो दैट्स वाई तो मैंने तो कुछ ज़्यादा ही बोल दिया, इतना तो बताना भी नहीं था, बट एनीवेज यहाँ तक हो गया हम लोगों का. देन वी आर the next topic is insertion of recombinant DNA into the host cell organism. There are several methods of introducing the ligated DNA into recipient cells. The recipient cells, after making them competent to receive, take up DNA present in its surrounding. So if a recombinant DNA bearing gene for resistance to an antibiotic, ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin resistant cells. If we spread the transformed cells on agar plates containing ampicillin, only transformants will grow. Untransformed recipient cell will die. Since due to ampicillin resistance gene, one is able to select a transformed cell in the presence of ampicillin. The ampicillin resistant gene in this case is called a selectable marker. Obtaining the foreign gene product. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial, plant, or animal cell, the alien DNA gets multiplied. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. Hence, there is a need for recombinant DNA to be expressed. The foreign gene gets expressed under appropriate conditions. The expression of foreign genes in host cells involve understanding many technical details. After having cloned the gene of interest and having optimized the condition to induce the expression of the target protein, one has to consider producing it on a large scale. Can you think of any reason why there is a need for large scale production? If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. The cells harboring cloned gene of interest may be grown on a small scale in laboratory. The cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. The cells can also be multiplied in a continuous culture system wherein the used medium is drained out from one side while fresh medium is added from the other to maintain the cells in their physiologically most active log slash exponential phase. This type of culturing method produces a larger biomass leading to higher yields of desired protein. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors where large volumes like 100 to 1000 liters of culture can be processed was required. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, etc. Using microbial, plant, animal, or human cells, a bioreactor provides the optimal conditions for achieving the desired products by providing optimum growth conditions like temperature, pH, substrate, salts, vitamins, oxygen. The most commonly used bioreactors are स्टेयरिंग टाइप विच आर शोन इन फिगर 11.7. यहाँ पे दो तरीके के बायो रिएक्टर है. सिंपल स्टर्ड टैंक रिएक्टर है एंड देन वी हैव स्पार्ज्ड स्टर्ड टैंक बायो रिएक्टर थ्रू विच स्टेराइल एयर बबल्स आर स्पार्ज्ड. तो एक तो नॉर्मली है जहाँ पे हम लोगों का ये पहला वाला जो है जो कि नॉर्मली है. यहाँ पे हमने मोटर लगा दिया है, फिर ये ब्लेड्स लगा दी है ताकि फोम अगर बन रहा है थ्रू आउट द प्रोसेस, क्योंकि हम क्या कर रहे हैं जो हमारा जो जेनेटिकली मॉडिफाइड ऑर्गेनिज्म है उसको हमें लार्ज स्केल पे बनाना है ना. तो हमने यहाँ पे क्या किया है कि हमने सिंपली यहाँ पर ऐसा एक बायोरिएक्टर एक वेसल जैसा ले लिया जिसमें कि हमने क्या कर दिया, हमने ये कल्चर ब्रॉथ वही है जो हम लोगों का जेनेटिकली मॉडिफाइड ऑर्गेनिज्म जो है उसको कल्चर ब्रॉथ इसी को बोल रहे हैं, उसको हमने डाला इस बायोरिएक्टर में. फ्लैट प्लेट क्या करेगी, जब मिक्स कर रहे हैं हम इसको तो फोम को ब्रेक करेगी. यहाँ पे मोटर लगा दिया. हमें एक एसिड बेस एक पीएच मेंटेन करना है तो हम एसिड बेस और पीएच कंट्रोल डाल दिया. स्टीम फॉर स्टेरिलाइजेशन कि कोई और ना आ जाए जो हमें डिज़ायर्ड जो ऑर्गेनिज्म है उसको ही सिर्फ क्या कहते हैं कि ज़्यादा उसका ग्रोथ करवाना है. तो स्टीम स्टेरिलाइजेशन के लिए डाल दिया. मोटर हमने इसको घुमाने के लिए लगा दिया. ब्लेड्स जो है वो हम लोगों का फोम को ब्रेक करेंगे एंड कल्चर ब्रॉड डाल दिया. नीचे से स्टेराइल एयर आ रही है, ठीक है. फिर यहाँ पे जो दूसरा है जहाँ पे स्पार्ज स्टर्ड टैंक बायोरिएक्टर बोल रहे हैं, यहाँ पे हम ऑक्सीजन का यूज़ करें. कुछ कुछ ऑर्गेनिज्म होते हैं जिनको ऑक्सीजन चाहिए रहता है उनके ग्रोथ के लिए, तो हम यहाँ पे ऐसा कुछ हमने एक पूरा वो सिस्टम क्रिएट किया जहाँ पे हम क्या कर रहे हैं कि ऑक्सीजन को ज़्यादा से ज़्यादा प्रोड्यूस करें. इंक्रीज सरफेस एरिया फॉर ऑक्सीजन ट्रांसफ़र. देन बबल्स ड्रामेटिकली इंक्रीज द ऑक्सीजन ट्रांसफ़र एरिया. तो बबल्स से और ऑक्सीजन बढ़ रहा है, ठीक है. और इसको हम घुमा रहे हैं पूरे को ताकि हम लोगों का ज़्यादा से ज़्यादा जो हम लोगों का डिज़ायर्ड ऑर्गेनिज्म है वो बने और हमें अपने ज़्यादा डिज़ायर्ड प्रोडक्ट मिले, ठीक है. अ स्टर्ड टैंक रिएक्टर ठीक है. Reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor content. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that bioreactor has an agitator system. Agitator यानी जो उसको ऐसे घुमाएगा, ठीक है. एंड ऑक्सीजन डिलीवरी सिस्टम, यहाँ पे ऑक्सीजन भी डिलीवरी सिस्टम है. एंड अ फोम कंट्रोल सिस्टम, अ टेम्परेचर कंट्रोल सिस्टम, pH control system, and sampling board so that small volumes of the culture can be withdrawn periodically. Downstream processing. After completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. The processes include separation and purification, which are collectively referred to as downstream processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo through the thorough clinical trials. As in case of drugs, strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. The summary आ गई है. Biotechnology deals with large scale production and marketing of products and processes using live organisms, cells or enzyme. Modern biotechnology using genetically modified organism was made possibly only when man learned to alter the chemistry of DNA and construct a recombinant DNA. This key process is called recombinant DNA technology or genetic engineering. This process involves the use of restriction endonuclease DNA ligase appropriate plasmid of viral vector to isolate the and ferry the foreign DNA into host organism, expression of the foreign gene, purification of the gene product that is the functional protein, and finally making a suitable formulation for marketing. Large scale production involves use of bioreactors. So thank you guys यहाँ तक सुनने के लिए और बाय.